So in this last section for this week, we want to discuss key features of scientific experiments. So what types of things did you notice about Hans Selye's work? I know you can't answer me here, but think about it just for a minute. Right? He was using rats. They were in a very confined situation, right? So he could control their living quarters. He could control how much they're fed, how much stuff they have inside their living space. Do they have anywhere to hide? Do they have a running apparatus? He could control all of their living accommodations. So controls in scientific experiments are very important, or controlling the variables, right? You want to have a test variable, and you want to have all of the other conditions be the same for both groups of rats. You also need the test subjects and you need the control subjects, right? Hans Selye wouldn't have found out any of this if he didn't have control subjects that he was only injecting with saline solution. He would have made the conclusion that the ovarian extract was causing those changes if he didn't have a control group that was getting saline solution alone. So you have to have a test variable that you're testing, and you also have to have a control for that test variable where you're not using that test in the same population. And then all of the other variables that can happen have to be controlled so they're the same in both groups. So those are some of the keys in scientific experiments that we learned from Hans Selye. What are some others? All right, so many of you may have learned about the scientific method, right? And so the scientific method can be used to design experiments and test hypotheses. Right? So you might start off with a research question. In the case of Hans Selye, he said, well, I have this ovarian extract and I want to know what it does to a biological organism. How does the ovarian extract affect the rat? So maybe he generated a hypothesis or maybe he didn't, right? He could have said, I think that the ovarian extract will cause the rats to maybe go into heat and go into reproduction. We don't really know what his hypothesis was during this test. Then he does the experiment. He injects the rats every day for months, and then he's got the control group that he's injecting with saline solution, and he gets his results and he draws his conclusion. His results in this hand, what he saw was that all of the rats got things like peptic ulcers, they had lower immune response, and they had atrophy of immune organs, right? So in this case, his hypothesis that maybe they would go into breeding phase or reproductive phase was not supported by this study. It would go into this side rather than this side where the hypothesis is supported, you report the results. Hypothesis not supported, you have to go back to the research question and say, what's going on here? Once you figure that out, then you can report the results. Sometimes you may also just want to report the results if your hypothesis is not supported so that other researchers know that you didn't find anything that connected those hypotheses to that experiment. So what exactly is a hypothesis? A hypothesis, by definition, is a supposition or proposed explanation made on the basis of some limited evidence that's a starting point for further investigation, right? So it's essentially the researcher's best guess why some phenomena occurred. So at the beginning here, right, you might make some observation, right? And you might say, which type of fertilizer works the best? And you might make a hypothesis. And you might say, plants grown with fertilizer A will grow the fastest. And maybe it's because that one had the most nitrogen, right? And so you say, okay, that's my hypothesis. The fertilizer with the most nitrogen is going to give the best plant growth. And so then you plant your plants, you use your different fertilizers, and you look at the results. If fertilizer A did cause the best plant growth, then your hypothesis was proven correct and you can make that conclusion. But if it's not, then you have to go back to the drawing board. And the one thing here that's not shown is that there are only 3 plants here and there's 3 fertilizers. You would need to do a lot of replicates as well so that you're sure you didn't make any mistakes, right? Mistakes happen. We're all human. So what if she forgot to add fertilizer to plant B, or she forgot to water it for 1 week, but watered everybody else? That's going to cause a big difference as well. So you need to have replicates done at different times and show that you get the same results. So how does a scientific hypothesis differ then from a scientific theory? Because in general terms, we use theory to actually mean a hypothesis. We might say, oh, my theory about why that happened is this. But that's actually not a theory. That's a hypothesis. Theory and hypothesis differ from one another. A scientific theory, by definition, is an explanation of an aspect of the natural world that can be repeatedly tested and verified in accordance with the scientific method using accepted protocols of observation, measurement, and evaluation of results. So theories are actually strongly supported in science. Something doesn't achieve the status of a theory unless there is a lot of data that support it. Something like the theory of gravity, for example. All this data, all of this mathematical calculations that have been done have shown that the theory of gravity exists, right? Or evolution. So all of the data that we've compiled to date about how species evolve is supporting evolution by natural selection, the way that Darwin described it. That there are genetic mutations that lead to variation and can cause different traits within species. So where possible, theories are tested under controlled conditions in experiments. And then they're supported by multiple experiments, not just one. So that's the difference between a scientific theory and a hypothesis that's generated by a researcher. So now let's talk a little bit about the types of reasoning that you can use when you're thinking about science. You can have the pathway that we just described, where you have some observation. You make some generalizations or predictions. So these would be your hypotheses. You do your tests, and as other people do their tests as well, then you can lead to the generation of a theory. This is called inductive reasoning. This is different from deductive reasoning, right? Deductive reasoning goes the other way. Now that I have this theory, right? The theory of evolution, for example. That theory is going to make predictions, right? And it's going to allow me to make predictions based on what I think I should see if this theory is actually correct. And then you can also design experiments to test that theory. So that's what's been done in the case of evolution. Right? Darwin made the observations, made generalizations, and then he surmised that natural selection was the way that species evolved on our planet, right? And so once you have that defined theory, now we have natural selection up here. We can make predictions from that, the way species should be evolving, and we can design experiments to then test the theory. And so both of these processes of reasoning in scientific experiments are very important, right? Because sometimes we don't totally get things right. Science is also evolving, right? We have different equipment that we develop. We can measure things more precisely. We get new math. We get new physics, right? We find out, oh, wow, there are more parts inside an atom than just the electrons and the neutrons and the protons. Who knew? Well, we didn't know until we could measure it. Both inductive and deductive reasoning are very critical, right? You might start with this inductive process, but you can't stop testing your theories. Nothing is ever proven in science, it's only supported, right? So your theory is supported by all this data, right? But maybe there's some piece of it that you didn't quite get. And so you're making predictions, and then this experiment doesn't turn out quite the way you expect. And you're like, well, why is that? And then you revise, your theory a little bit based on those experiments that may tweak it, right? And you get the theory of relativity instead of just Newtonian gravity and predictions. Okay, so what we've learned today, scientific reasoning needs to be both inductive and deductive. And so we will see how this type of scientific reasoning lends itself to the medical field and how we use it in Western medicine. We also learned about the scientific method, looking at the development of hypotheses, how that can be tested using experimentation, and then over time developing theories based on our repeated observations that support that theory. And that is where we will end for this lecture.